Okay, thanks, Vic. Good morning, everyone. I'm Andrew Muir, and I'm going to present to you a project that we've been involved in over the last few years on the ciscoes of the Laurentian Great Lakes and Lake Nipigon. And for the purpose of this talk, I'm not going to focus on Lake Nipigon just because of the time available. So much of what we know about the diversity in terms of the taxonomy, the systematics, and the ecology of ciscoes in the Great Lakes is based on this seminal work by Walter Kelts, published in 1929. It was part of his doctoral work. It was completed in the 1920s. He examined roughly 9,000 specimens that were collected between 1917 and 1924, so roughly a century ago. He painstakingly collected 32 morphometric and meristic pieces of data on variables for all of those fish. In the monograph, he describes 9 species, 4 of which he named, as well as 12 subspecies. So Kelts' monograph is a seminal piece of work, and it's relevant today, but it's complicated, and as I mentioned, it's somewhat outdated given that these collections are a century old. There also have been several extirpations and extinctions. There have been changes in taxonomy over time. There have also been changes in morphology within forms and among lakes. A couple of potential caveats to the Kelts' work is that there was no statistical treatment of the data. And a single dichotomous key was presented for all forms in all lakes combined, and that's problematic, as many of you will know if you've tried to apply that key to identifying forms. It doesn't recognize the variation within forms among lakes. So the objectives of my presentation today is first to provide you with an updated status of the ciscoes in each lake, to identify notable changes in morphs over time, and I'll explain what I mean by that, and then to present to you some questions and some recommendations on management and research as the lake committees become more engaged in cisco restoration in the Great Lakes. So what's the relevance of this and the timeliness of this? Well, there are 2 primary strategies. I'm going to present to you the 2 ends of the spectrum here with respect to reestablishment. The first is to stock a locally adapted form into suitable habitat in the receiving water body. The second strategy is to stock a less differentiated, more genetically diverse form into the open lake environment, thereby mimicking early stages of evolutionary divergence. So you're putting in the potential variation that exists in adapted forms. So there are positives and benefits to each end of that spectrum, and that strategy will differ depending on whether we're talking about shallow water Coregonus artidi versus the deep water cisco forms. So just to set a bit of context there. Some of the basic concepts I want to introduce before I get into the results is that historically, as we know, Kelts named these forms species, so giving them a proper genus and specific name, as well as subspecies, so things like Coregonus artidi albus. These are the subspecies. Albus and Manitolinus are the primary ones. We deal with them as forms. We refer to them, as you'll see on the right-hand side of this figure, these are 5 of the primary forms, the main forms, but we call them by their specific name without the genus name, only in the colloquial sense in this guide, and recognize them as forms rather than distinct species. And we also describe 2 minor forms, the albus and the Manitolinus form. Second concept here is that historically the entire basin has been considered to contain a species flock. We regard each lake as containing a species flock, and that recognizes the variation within those forms among the lakes. So our work is based on 3 different data sets from 3 different time periods. And I mentioned here at the top morphometric and meristic data. What I mean by morphometric data are things that are measured. So linear distances shown on the bottom right-hand side of this slide. Things like eye diameter and fin lengths are morphometrics. Meristics refers to count data, so things like gill raker numbers. So the first data set, as we already saw, is from Celts from the 1920s, roughly about 9,000 fish. A second data set that we spent a lot of time digitizing for this, well, we didn't, other people did spend a lot of time doing this. From the 1950s to the 70s, those were collections made by Stanford Smith, representing roughly 1,400 individuals. And then the contemporary data from the 2000s onward, roughly 3,300 fish. So in total, we have about 13,000 individual fish that are represented in this field guide. So a huge amount of data supporting our analysis. The guide is organized around 8 morphometric and 1 meristic character, Gilraker numbered, that are common to each of those 3 data sets. So we can detect changes over time in those variables. And we have those for all of the extant forms in all of the lakes. We recognize the 8 major forms and 2 minor forms. These are organized by lake, so in contrast to Celts, we separate them out by lake so we can recognize and understand that variation and differences among the lakes. It's well illustrated thanks to our colleague Paul Vecchi. We've got about 30 color illustrations in the guide. We can, as I mentioned, detect morphological changes from the 1920s through to contemporary times. And we provide a very detailed overview of the taxonomy, the distribution, and the ecology of these animals. And you may wonder why morphometrics and meristics are important. And part of that is because currently we don't have other means of identifying these different forms. Also, a lot of these morphometric and meristic variables are adapted to specific environments and also specific trophic resources that these animals are using. So things like eye diameter are adapted to living in deep versus shallow water environments, or preying on organisms from above versus from below. So these are not just markers of change, these are adapted variables that we're considering here. And some of those things need to be considered as we try to identify source populations for reintroductions into particular habitats. So the next slides, I'll get into some of the results, and I'm just going to give you a sense for what the guide looks like, and then I'll talk more specifically about some of the key results for each of the lakes. So we provide an overview for each of these main forms with illustrations, and we compiled historical data, so pre-2000s, on the distribution of each of these main forms. So shown in red here is the historical distribution on the left, and in right is the contemporary distribution. And the contemporary data is based on not just on published information, but also a lot of data from the USGS trawl surveys was compiled here. So we provide an update on that. This is an example of the Lake Superior Cisco Clock, just to give you a sense for some of the illustrations that Paul has done for the guide. So the next few slides here, I'll present to you the lake-specific information, and we tried to focus on outcomes that were particularly relevant to management. So this table here shows the major forms on the left-hand column, the first column, and the lakes across the first row. And I want to highlight a few things. So these boxes in blue represent the extant species or the extant forms in each of the lakes. You can see Lake Superior and Lake Nipigon primarily contain the forms that were historically extant. These boxes in yellow represent both extirpated and extinct forms. One thing to note is that Michigan and Lake Ontario have been particularly affected over the past century. The boxes in green here represent fishes for which we are uncertain of their status, so I'll talk a bit more about that in coming slides. And then finally, in pink, I guess this is, Lake Huron, we recognize that the deepwater ciscoes may now be intergressed into a species flock, and that was alluded to by Tom Todd and others in the 90s. So I'll talk a bit more about that as I get into each lake. So each of these next slides provides some of the results by lake. So for Lake Superior, as we just saw, there have been no extirpations in the last century. However, contemporary Artidae are showing 3 fewer gill rakers than the historical collections, and this change has occurred since Smith did his collections in the 50s. What that means in terms of the biology and the ecology of the organism, we're not exactly sure right now, but that's considered a notable difference from historical times. I mentioned that Negropinis and Regardia, the status of those 2 forms is uncertain in the lake. We do have some records that suggest that those forms may indeed be present in low numbers. We have some data from an analysis by Dan Ewell showing that the albus and manitolinus minor forms may in fact occur in Lake Superior, and manitolinus was never historically recorded in that lake. With respect to Lake Michigan, a bit of a different story. We've had 6 extirpations, as you know. Artidi and Hoyai are the only 2 extant forms remaining in the lake. For Artidi, we note 5 of 8 body metrics for contemporary Artidi diverged notably from those measured by Celts. And so I'm going to revisit that point here when I summarize. 3 other metrics diverge marginally. So on the basis of our analysis, some pretty significant changes in the form and potentially function of Artidi in this lake. Hoyai, on the other hand, we note only one difference. That is, pelvic fins appear to have shortened. And oddly enough, pelvic fins have shortened in all of the extant populations of Hoyai in all of the lakes. We don't know what that means, but it's interesting that it occurred in all of the populations. We also note temporal variation in gill raker numbers. In Lake Michigan, there was a period during the 80s when Crowder and Crawford did some work where gill raker numbers dropped considerably. They seem to have gone back up to what they were in the 50s in the contemporary data set. For Lake Huron, we identified that an albus-like form is indeed extant, and Steve, if you were here on the first day, Steve Leonard mentioned that they've been sampling the Lachinoes and they have what's considered an albus-like. So albus is the deeper bodied form of Coregonus artidi. Those appear to be extant, but we don't yet know the status of typical artidi, those offshore pelagic spawning artidi in Lake Huron. 4 of 8 body metrics for contemporary artidi diverge notably from those recorded by Celts, so half of those. Based on the morphological data, we're providing some pretty, I think, convincing evidence that the deep water forms have all introgressed into a hybrid swarm, and that likely occurred sometime after the 1950s. The hybrid that exists now is somewhat more Hoyai-like, but the morphology is intermediate between all of those pre-existing deep water forms, of which there were 5 prior to the 1950s. In Lake Erie, we've seen no evidence of remnant albus. Albus in Lake Erie was the dominant form, or typical artidi. There have only been 29 ciscoes collected since 1995. Randy Eschenroder had a chance to look at data from those 20. Our co-author Nick Mandrak worked those fish up and found that of those animals, 19 of them are morphologically similar or comparable to the hybrid form in Lake Huron. So it's likely that those animals moved downstream in the system. In Lake Ontario, we've had 3 extirpations. However, typical artidi is almost identical morphologically to the animals that were collected by Celts in the 1920s. So very stable morphologically. Relatively few Hoyai have been reintroduced into Lake Ontario, as you know. So I say relatively few in comparison to what the historical population may have been, but there have been tens of thousands of animals introduced into Lake Ontario. So just to summarize those key results, there have been 21 instances of notable change, and we define that as 2 standard deviations difference from the mean collected of the data collected by Celts in the 1920s. The rate of change, if you look at it on a per form basis, it varies between 0.2 and 0.25, so that's fairly uniform across the forms. Some forms, as we saw for Lake Ontario artidi, have been extremely stable morphologically. Artidi accounted for nearly half of the notable changes across the lakes. However, one thing we're working on now is that we're realizing that we may indeed not be comparing the same animals, because Keltz didn't sample a lot of these nearshore populations in his data set, and so we may indeed be comparing the offshore pelagic typical Artidi form to things like albus or other forms. So we're working on that right now. One thing is for certain: if Keltz had sampled, for example, the Grand Traverse Bay population, it's almost certain that he would not have called those typical Artidi. They just differ dramatically in their morphology. And the same would probably be true of some of those Les Chenaux populations. So just to kind of wrap up, I'll present to you a few questions and some potential management needs that we may want to consider. Do we need a coordinator to provide quality control and coordination of these efforts and data as the managers begin to more fully engage in restoration efforts across the basin? We recognize the need to train field biologists in morphometrics. We're not recommending that morphometrics be done on all of the assessment collections. However, when we're trying to identify remnant stocks or animals that may prove to be good sources for reintroduction, morphometrics come in handy. We need to develop management-based assessment priorities for each of the lakes. So some of the examples could be, is albus extant in Lake Huron? What's the status of typical Artidi in Michigan and Huron? And John is working with the CLC to organize a workshop this coming fall, really to start to discuss some of those management priorities for the lakes. And we envision that the science that supports those management priorities will be developed thereafter. We need to understand the data gap for juveniles. So all of the existing efforts focus mainly on adult-sized fish, greater than 200 millimeters. Animals that are stocked into Lake Ontario are going to reproduce, hopefully. If we're successful, we need to be able to identify juveniles. We need to fill in some data gaps in existing collections. There's a large set of animals that are deposited at the University of Michigan Museum of Zoology, collected by Stan Smith, that have never been given accession numbers. So they're there, we just can't access them and use them, and a lot of that data still remains on IBM punch cards. So there's some potential work that can go on there. We need to identify stock structure for all forms and the behavioral basis for stock structure in things like Artidi that were thought to be a pelagic offshore spawning species. And finally, assess plasticity in Artidi, replicating some of the work that Tom Todd started and did in the 80s through breeding experiments or common garden experiments. And so with that, I'll just mention that publication of this guide is expected during the summer of 2016. This is only possible due in a large part to the work that Randy has dedicated to this over the last couple of years. He's done a huge amount of the work, the bulk of the writing, and he's been instrumental in getting this to where it is today. So thank you, and I'll take any questions. I have maybe a minute or so here, it seems.